Good evening. Thanks for joining. We will begin in just a few moments. We just want to give everyone a chance to log on. Welcome. Thanks everyone for joining. All right, let's go ahead and advance to the next slide. Hello everyone, thank you for joining us for tonight's webinar as we share an overview of gene therapy, along with updates from Tenaya's clinical trials. My name is Nikki Butler, and I'm the Director of Digital and Social Media for the Dilated Cardiomyopathy Foundation and the Genetic Cardiomyopathy Awareness Consortium. We're so glad you can be with us as we explore advancements in gene therapy for inherited cardiomyopathies. We have over 350 registrants for this informative webinar. Next slide, please. I have just a few housekeeping items. All attendee microphones are on mute upon joining so that you can hear our presenters. Please enter your questions into the Q&A feature of Zoom at any time. There will be a dedicated question and answer session following Dr. Tingley's presentation this evening. And next slide. We do have a poll for tonight. So Justine, if you can push that poll number 1 forward. We'd love to know who's attending tonight's webinar. Are you best described as a cardiomyopathy patient, a family member or a friend, a medical professional, or an industry professional? All right, let's see here. Everyone a chance to answer. All right, we have about half our cardiomyopathy patients, 20% industry professionals, 18% family member friends, and 10% medical professionals. Thank you. I can share that. It is always interesting to see those results. So let's keep rolling. I do want you to know that this webinar is being recorded and will be posted on our YouTube channel following tonight's discussion. As registrants, you will receive an email with a link to the recording. Our YouTube channel also houses a library of our previous webinar recordings, and using the auto translate feature, these videos are available in 54 different languages. Next slide. To begin, I'd like to give you a little background. The DCM Foundation, focused on dilated cardiomyopathy, created the Genetic Cardiomyopathy Awareness Consortium, or GCAC, in 2023. This effort began in 2022 when the DCM Foundation recognized a major gap. Genetics in cardiomyopathy wasn't being widely discussed, not by medical professionals and certainly not by patients. And we realized something important. You can't talk about genetic cardiomyopathy in silos. Many gene mutations cross diagnostic boundaries. For example, a single gene variant might cause dilated cardiomyopathy in one person and arrhythmogenic cardiomyopathy in another. Because of that overlap, we needed a broader, more inclusive approach. So GCAC was formed to raise awareness and provide education about genetics in cardiomyopathy across all types. Next slide, please. In just a few years, we've made very meaningful progress. Awareness of genetics in cardiomyopathy has increased significantly, both in the medical community and among patients. When we launched GCAC, we started with 8 patient groups. Today, we've grown to 11, and we are continuing to expand. This momentum shows there's a real need and real interest in understanding the genetic side of cardiomyopathy. Next slide, please. The 11 patient groups that are participating in the Genetic Cardiomyopathy Awareness Consortium, along with the DCM Foundation, are the Children's Cardiomyopathy Foundation, Laminae Cardiac, the Danon Foundation, Mended Hearts, PLN, SADS Foundation, Women Heart, Let's Talk PPCM, Team Titan, Heart Charged, and the Sudden Cardiac Arrest Foundation. We want to thank each group for participating. It's great to have a coalition of 11 different groups working together to solve a real problem with patients. And next slide. Due to an unexpected conflict, Dr. Matt Pullman is unable to join us this evening, but that said, we are grateful to welcome Dr. Whit Tingley, Chief Medical Officer at Tenaya Therapeutics. Dr. Tingley brings extensive leadership in cardiovascular research and clinical development with prior senior roles at Genentech, Roche, and Cytokinetics. He is a former adjunct associate professor of medicine at the University of California, San Francisco, where he also practiced as an attending cardiologist. Dr. Tingley earned his undergraduate degree from Brown University and his MD and PhD from Johns Hopkins, with postgraduate training at Johns Hopkins, UCSF, and the Gladstone Institutes. Dr. Tingley, we're honored to have you with us tonight, and we look forward to your insights. So I'll turn this over to you now.
Thank you, Nikki, and thanks to the DCM Foundation and GCAC for hosting, and most importantly, thanks to everyone that has joined and is listening. I look forward to describing our progress with gene therapies for two forms of genetic cardiomyopathy, and welcome your questions at the end. So please save your questions. I look forward to that part most. Next slide, please.
We're just having that same problem. Just give me 1 second here.
No worries. The next slide will have the agenda. I'll give the highlights from the trial results first, and then we'll go into a deeper dive on ARVC caused by mutations in the PKP2 gene.
I'm going to have to just take this down. So give me 1 second here.
Yep.
Apologies.
And then I'll do a little bit of a interim on how gene therapy works. And then we'll go to hypertrophic cardiomyopathy, the MYBPC3 gene, and our trial there, which is called MyPeak-1. And then, of course, the questions. All right, next slide. Next slide. So at Tenaya, we are all about science for patients and putting patients first. Next slide. We are 100% focused on heart disease. We have 2 active gene therapy studies in hypertrophic cardiomyopathy and ARVC that we'll cover today. We aren't talking about today, but we also have 2 important observational studies. An observational study means we don't have a treatment we're testing or an intervention, but we are following patients, collecting data to much better understand the natural history of disease. We do everything with patient input and patient needs in mind. We have a dedicated advocacy function. We rely heavily on our patient advocacy council that gives us feedback on our protocols, on our materials for the trials and so forth. We take education very seriously, and we would love to hear from you. So our email is patient.advocacy@tenayathera.com. Next slide. All right, this is the news, the highlights from both of our gene therapy trials. In ARVC, we have RIDGE-1. We've enrolled 6 patients with PKP2 mutations into that trial. We have tested 2 levels, 3 patients at each dose, and TN-401, the gene therapy, has been tolerated at both doses. We do take heart biopsy samples to see if the gene therapy is doing what it's supposed to do in the heart. We measure DNA, RNA, and protein, and we are encouraged by what we're seeing. We'll go into more detail. Most importantly, the first 2 patients enrolled who have the longest follow-up period, the longest time since they were treated, are showing signs of improving electrical stability in the ventricle of their hearts. Moving forward, our next step is a data safety monitoring board. That's an independent group of people who review the data coming from the trial and determine and endorse whether it's appropriate to proceed and dose more patients. Once we get through that, we anticipate resuming enrollment in the RIDGE-1 trial, all as planned per protocol. On the hypertrophic cardiomyopathy side, we have the MyPEAK-1 trial. We've enrolled 7 patients with mutations in the MYBPC3 gene. Again, it has TN-201 in this case has been tolerated at both doses. The heart biopsies are showing that it's doing what we expect in terms of DNA, RNA, and protein production. We have been able to successfully adapt the protocol to reduce the amount of immunosuppression used. So these gene therapies do require a temporary use of medicines to quiet down the immune system so it doesn't react to the gene therapy. We have a longstanding goal to reduce the amount of immunosuppression needed, and we're making progress there. Importantly, patients in that study are feeling better, and we'll talk about the New York Heart Association class improvements in particular. And then pictures of the heart and blood tests are showing signs of improvement in the heart as well. All right, next slide. We'll transition to ARVC and the PKP2's role in ARVC. Next slide. So ARVC most commonly is caused by disrupted connections between the cells in the heart. Without good connections, cells can pull apart, and that can disrupt their electrical connections as well. And of course, the electrical connections, that's what makes the EKG. That is necessary for normal function of the heart. When these connections become disrupted, it can lead to short circuits or abnormal heart rhythms, arrhythmias that can be life-threatening. Also, as the cells pull apart, it can become scarred, and those scars in the heart can add up and eventually disrupt the ability of the heart to pump blood. Now, there are many genes that can cause ARVC. PKP2 is the most common genetic cause. PKP2 makes a protein that acts like a Velcro to hold the cells together and to hold the electrical channels in place. TN-401 is our gene therapy. It delivers a full-length copy of the normal PKP2 gene sequence to allow the cells in the heart to resume producing PKP2 protein and build back those connections. And RIDGE-1 is the name of our ongoing clinical trial to test the activity of TN-401. Next slide. There are no therapies currently that address the underlying genetic cause of PKP2-associated ARVC. There is, of course, standard of care, which is very helpful, but not enough. Implantable cardiac defibrillators are key and are life-saving, but they're painful, traumatic, they have their own complications, and they go off sometimes inappropriately. Not ideal. Antiarrhythmic medications do work for some patients, or they work for a period of time in some patients, but they are certainly not fully effective, and they have their own side effects. There's a surgical procedure called ventricular tachycardia ablation. This can have high short-term success in the most experienced centers, but the long-term success is more variable. Another part of standard of care is exercise restriction. Many people with ARVC are athletes or, unfortunately, former athletes, because once you have ARVC, you need to stop high-impact aerobic exercise. So we need a new treatment paradigm to prevent disease onset, to stop disease progression, and to reverse disease once it's been established. All right, the next slide, 10, is a little mini medical school on what causes life-threatening heart arrhythmias. So there is a range of abnormal heart rhythms that come from the ventricles, starting with premature ventricular contractions. Those are single beats. then runs, short runs called non-sustained ventricular tachycardia or NSVT, then sustained ventricular tachycardia, ventricular fibrillation, and sudden cardiac arrest. So sudden cardiac arrest, when people suddenly pass out, are typically caused by ventricular fibrillation or ventricular tachycardia. Okay, what's going on inside the heart? So a normal heartbeat starts in the right atrium where that arrow points. It forms a little bump on the EKG where the other arrow points, a little round bump, and we call that the P wave. That's the sign that the normal pacemaker started that beat. And then the beat conducts through the ventricle, and it does so quickly and efficiently. And so the ventricle electrical signal is that sharp spike. And importantly, it's really narrow, which means it's happening quickly. So that's what a normal heartbeat looks like. Now, where the star is, that could be a short circuit, like a spark, a sudden spark that causes an extra beat. It starts in the ventricle and it spreads more slowly. So the PVC is a wide complex, and you can see that single wide beat. That's the PVC. Now, as disease progresses and fibrous tissue forms, it can form short circuits that run electricity continuously, like the wires stay touched, not a spark, but the wires remain connected. So that forms a short circuit that goes around and around in a loop. We call it reentry. And that can form non-sustained VTAC, so multiple wide beats together that resolve spontaneously, or a sustained VTAC if it doesn't resolve spontaneously. And VTAC can be at a variety of heart rates. Some are still pumping blood and people feel their heart racing. They might feel lightheaded. And some may be too fast to effectively pump blood. Patients will pass out. And then this can degenerate into ventricular fibrillation. That's the very wavy waveform there. No blood is being pumped. That can cause sudden death, unfortunately. So on the good side, the VTACs, the VFibs are rare, but of course, very dangerous. On the flip side, PVCs aren't directly dangerous, but they happen a lot. Patients with ARVC can have hundreds to thousands a day. In our trial, you have to have at least 500 a day to get into the trial. Non-sustained VTACs are less frequent than PVCs, but there's a next step on this causal pathway towards life-threatening arrhythmias. Next slide. So we can use the PVCs and non-sustained VTACs in many ways. They are a hallmark of ARVC, but they do occur in many other diseases, and some people with healthy hearts also get PVCs. But high PVC counts in ARVC mean a high risk of life-threatening arrhythmias. And the number of PVCs and the presence of non-sustained VTAC are used in the calculations that doctors and patients make to determine which ARVC patients should be treated with an implanted cardiac defibrillator, or ICD. The goal of TN-401 gene therapy is to stabilize the electrical aspects of the ventricle. We can most easily measure this by counting the numbers of PVCs and non-sustained VTAC. And since these are happening daily, these are going to be an early indicator of improving electrical stability in the ventricle. Ultimately, of course, the goal is to lower the risk of severe arrhythmic events overall. These events correlate directly with the number of PVCs. So we anticipate that reductions in PVCs and non-sustained VTAC will result in lower risk of life-threatening events. Next slide. All right, with that, we'll transition into early results from the RIDGE-1 study of TN-401 gene therapy for PKP2 ARVC. Next slide. This study, the objectives include safety and tolerability. How does the body tolerate TN-401? We are looking to find the best dose, and we're looking for early signs of activity, what we call pharmacodynamics. The trial has 2 initial cohorts of 3 patients each, and after that it can expand to up to a total of 15 patients in the trial overall. Each of those first 2 dose cohorts has a different dose level. 3E13 vector genomes per kilogram and 6E13 vector genomes per kilogram. Excuse me. And I mentioned we have an independent group, the DSMB, that is meeting regularly to monitor the progress and the safety in the trial. So for a patient participating in a trial, what you find is first you sign informed consent. You work with the treating physician and the staff to learn about how the trial works, what the potential risks are, what the potential benefits might be, have all your questions answered. And if you decide to participate in the trial, you sign this informed consent. We go through a screening process to determine whether you're eligible to participate in the trial, and there are a variety of criteria. And then we do a variety of pre-dose activities or baseline assessments, we call them, to measure the state of your disease before the treatment. Gene therapies are a one-time treatment infused intravenously. Patients are followed up very frequently over the first weeks, then less frequently over the next months. And that kind of primary efficacy measurement point is 1 year after the first dose. And then patients enter 4 more years of long-term follow-up with much less frequent visits. The study does include heart biopsies, and we'll talk about the results from those. And it does use prophylactic immunosuppression, a temporary period of weeks to a few months to keep the immune system under control after the dose. Next slide. All right, so we have dosed all 3 patients in both dose cohorts at this point. Cohort 2, patient 4, 5, and 6, all were dosed relatively recently. We don't have data about the efficacy of TN-401 in those patients to share today. We look forward to learning more and sharing it next year. But the dosing has all been successful and it has been well tolerated to date. Cohort 1 has had longer follow-up since the dose, from 20 to 40 weeks at this point, so still early but meaningful follow-up. 3 men were dosed. The youngest was diagnosed at age 16. All 3 have severe enough disease to require implanted defibrillators, and they have all had the ventricular tachycardia ablation surgery procedure. One had a repeated one. But despite that, and despite standard of care medications, they continue to have symptoms and electrical instability in the heart with high PVC counts, as listed there. All of them are New York Heart Association Class 1, which means they don't have heart failure symptoms now. One goal of TN-401 therapy will be to prevent heart failure longer term. Next slide. We're very encouraged by the emerging safety profile of TN-401. It's been well tolerated at both dose levels. We have not had any dose-limiting toxicities to date. We have not, when we look at heart function, we have not seen any signs of heart damage with this, which is very encouraging, and we have not seen any evidence that the treatment increases arrhythmias. The majority of events have been that, due to the immune system, have been mild and easily manageable with the immunosuppression regimen, and all patients in the first cohort have successfully tapered off their immunosuppressive medications. Cohort 2 dosed most recently. We are in the progress of that taper now. Next slide. So we are very excited to see the emerging data in terms of electrical stability. So these the counts of premature ventricular contractions per day, both at screening, so before the treatment, and then the most recent visit for patient 1, 2, and 3. So it's excellent to see substantial drops in the PVC counts at week 40 in patient 1, a 46% drop, and an 89% drop for patient 2 at week 32. Patient 3 has not dropped at this point. They have the shortest follow-up of the group at this point, week 20. It does seem to take about 6 months for this gene therapy and other gene therapies for the heart as well to take effect. So we are cautiously hopeful that patient 3 will see similar reductions with more time. As I said, the frequency of premature ventricular contractions predict the likelihood of life-threatening events. And in epidemiologic studies, that's a linear relationship, which means that the first two patients who on average had a 67% drop in PVCs might have a reduction in life-threatening arrhythmias by as much as 67%. However, that's mathematical, that we have not proven that at this point. All right, the next slide talks about non-sustained VTAC. Patient 2 had a very high burden of non-sustained VTAC at screening before treatment, 78 per day. And I'll say that we measure this every day for a week. We count every heartbeat for a full week and then average it out to get the daily count. So 78 events, if you add that for a week, that's a lot of events. And of course, it was happening even before the monitor was put on. So we are extremely excited to see that patient 2 had 0 non-sustained VTAC events for the week ending on week 32 weeks after the dose. So very encouraging early results from this patient. Patient 1 and patient 3 have much lower burden of non-sustained VTAC at baseline. We wouldn't expect to see this big a signal in them. So encouraging initial data on improving electrical stability in patients in the first cohort. Of course, we need to learn more. We're very grateful for these patients and the next 3 who have already been dosed. We look forward to sharing longer-term data from all 6 of these patients in the first half of next year. And we look forward to additional patients participating in the study to help us understand the safety and efficacy even better. All right, well, that will move to the next section. Next slide, please. How is TN-401 working in the heart? Next slide, very brief primer on genetics and protein synthesis. So the genes make proteins. The instructions for the whole human body and how it works is spelled in 3 billion letters, DNA base pairs, A, C, G, and T. 3 billion letters, by the way, would be like a 50,000-page college essay. So that's a lot. Each cell has a copy of that in the chromosomes. And those letters spell words, essentially. The words are to make proteins. So the main role of all of this is to make proteins. And humans have about 20,000 different proteins. Different cells use different combinations of proteins, but they carry out all the main work of the human body. Next slide. Now, of course, if the proteins don't work or the proteins aren't there, the body won't work well. So different types of genetic mutations can have different effects on proteins, but the ones we're talking about today from IBPC3 and PKP2 are ones that just prevent the protein from being made. And so there's not enough protein, and the cell can't do what it would normally do without that protein, and that leads to cardiomyopathy. So TN-201 and TN-401 are designed to deliver a new copy of the gene to the heart muscle cells so that the protein can get made and the cells can do what they're supposed to do. Next slide. Now, we've talked about exactly how this works in more detail. We have less time today, and I do want to make sure there's plenty of time for questions. So I won't go through in detail how we deliver the gene to the heart cells, just to say that we use the proteins from a virus. The virus has evolved to deliver genes. We take out the virus's genes. We put in the MYBPC3 or the PKP2. It's a single intravenous infusion. Those proteins from the virus, the AAV, know how to get it to the heart and inside the heart cell to the nucleus. So the nucleus can take up the DNA. It uses the DNA to make messenger RNA, and then that is used to make the protein. So that's all I'll say, but if it comes up in the Q&A section, I'm happy to go into much greater detail about how it works. Next slide. So with the heart biopsy samples that we are getting, we look for the DNA, the RNA, and the protein in the heart, and we compare from before the dose to after the dose. And I'll say we see clear delivery of the DNA to the heart. We see production of the messenger RNA, and we know only the only the cardiac myocytes, the heart muscle cells can do that. So we know the muscle cells are getting the gene and turning it on. And then we do see increases in the protein. So this graph shows the PKP2 protein levels, the change from before the dose and after the dose. Patient 1 and patient 2 had, on average, a 10% increase in PKP2 protein since pre-dose. We use a very precise assay, and these are very real increases. Patient 3 did not have that increase. In fact, the assay said there was a slight drop, but that was in the range of variability of the assay. And so it basically means unchanged for patient 3 at this time point. This is measured early, 8 weeks after the dose. We know from our other trial that the protein continues to increase from 8 weeks to 1 year. And so we expect when these patients return for their biopsies at the 1-year time point to see larger increases and increases in all 3 patients. Next slide. All right, now what's happening inside the cell? So heart muscle cells are stacked like bricks, and they connect end to end on the long ends or the small sides. And that's where the electrical current goes through. The heart is very carefully designed to make sure the electrical current goes in the right direction between, across, and through the heart muscle cells. So there are these special channels that conduct electricity at the ends of the cells. And PKP2 holds those ends together like Velcro, and it holds those channels in place. If that's disrupted, the channels spread out and they can be more side to side, and that can create the short circuits that we talked about before, either the sparks, the PVCs, and set up for those reentrant arrhythmias, the non-sustained and sustained ventricular tachycardias. Next slide. So in the patient heart biopsy samples, we can take a look to see if the new PKP2 protein is going where it needs to go. So it's supposed to line up at these short ends. I have a schematic at the bottom here, the cells stacked like bricks with the yellow PKP2 protein at those ends. Those are called intercalated discs. And then the pictures here are pictures of a very small piece of patient one's heart, one taken before treatment with TN-401 and one taken 8 weeks after. And so this is just an example. We need a lot more data to prove changes at this level, but it is suggestive that the new PKP2 protein is getting exactly where it's supposed to go to help hold the cells together and hold those electrical channels in the right place. Next slide. So as I said, the next step is a DSMB meeting. Once we pass that, we will resume enrolling. The patients we're looking for, there are a long list of eligibility criteria, of course, but the key features we're focused on adults at this point. You have to have a PKP2 mutation, of course. You have to have at least 500 PVCs every day to get into the trial. You must have an ICD already implanted. And you need to have low levels of AAV neutralizing antibodies. We didn't have time to talk about it, but some people, maybe 20%, have antibodies that will bind the gene therapy and might prevent it from working. As you can see, we are well represented across the United States. We have also recently activated 2 sites in London. So if you are interested in learning more about the trials, you can contact us at the contact information at the bottom. All right, with that, I will translate, switch to hypertrophic cardiomyopathy. So the most common genetic cause of hypertrophic cardiomyopathy is MYBPC3, and our first in human trial is called MyPeak-1. Next slide. So There are two types of cardiologists. There's the plumbers and the electricians. Arrhythmic cardiomyopathies, like ARVC, you go to the electrician first because the first problem is often the short circuits, and then the pump function, the plumbing part, is a problem later in the disease. HCM and other forms of cardiomyopathy, it can go the other way, where the pump is affected first, but you can also get short circuits and electrical problems. In hypertrophic cardiomyopathy, it's clogged or too stiff to pump. So the mutations cause thickening of the heart muscle and it becomes inflexible. The most common genetic cause of hypertrophic cardiomyopathy is NYBPC3, and it makes a protein that is directly regulating the contraction itself. It regulates the proteins that cause the cells to shorten. But without it, the cells become thickened and they don't metabolize energy properly, and this ultimately results in disease. So TN-201 is designed to deliver a full-length copy of MYBPC3 to the heart muscle cells, make more MYBC protein, and restore normal regulation of cell contraction. And MyPeak-1 is the clinical trial. Next slide. I won't run through this slide in detail. It's very similar to what we talked about with the Ridge-1 trial. Next slide. So we have enrolled 7 patients total in MyPeak-1, 3 at the first dose level of 3E13 vector genomes per kilogram, and 4 more at the higher dose level. Overall, 6 women, 1 man, all have the non-obstructive form of hypertrophic cardiomyopathy. All are symptomatic and all have substantial hypertrophy on their echocardiograms. All 7 have severe enough disease that required implantation of an ICD. 4 of them used to have the obstructive form of hypertrophic cardiomyopathy and have undergone surgical myectomies to remove the extra heart muscle from their heart that was causing the blockage. So really quite severe levels of disease despite all the available therapies, including these myectomies in 4 cases. These patients continue to have symptoms, high risk of progression, and chose to participate in our trial. Next slide. TN-201 also has encouraging early safety profile. It's generally been well tolerated. There have not been no dose-limiting toxicities. There's been no evidence of damage to the heart. There is inflammation, as always, with gene therapies that has been manageable with immunosuppression. And all patients in both cohorts 1 and 2, so the first 6 patients have all successfully completed their immunosuppression. And as we've gone forward, we've been able to reduce the total amount of immunosuppression used to control the immune system. And we've taken that learnings across both trials now, both the MyPeak-1 and the Ridge-1 trial. Next slide. In the heart biopsies, we are seeing increase in protein. It was by patient 3, we started getting pre-dose or baseline biopsies, and you can see that patient between the pre-dose and week 52 or 1 year, they had a 5% increase in protein. And now the first patient from the higher dose, where we have a post-dose biopsy, that's patient 6, and we see a 14% increase in protein. So we're encouraged by the production of new protein and by the dose response, indicating that everything is working as expected. And the DNA and RNA levels in the heart biopsies do tell us that it is working as expected at the molecular level in the heart. The next slide talks about New York Heart Association class measurements. Many of you are probably familiar with this, but it's a measure that the investigator makes after interviewing the patient about their symptoms and how they're limited by their symptoms. So class 1 is the best. You're unlimited by your heart symptoms. You can do all ordinary activities. Class 4 is the most severe, where you have symptoms even at rest without activity. Now, in my peak 1, before treatment, all patients were either class 2 or class 3. We saw improvements, and by week 40, all 4 patients had improved to the best class, class 1. And then a subgroup of patients have been followed further, 52 weeks for 3 patients, 72 weeks for 2 patients, and they have all remained stable at class 1. So very encouraging early data about patients' symptoms and their limitations. Next slide. The heart imaging is also encouraging. So we are focusing on 2 different ways of measuring hypertrophy. Hypertrophy just means growth, thickening, or enlarging. So on the pictures of the heart, we can directly measure the thickness of the wall, and we can measure it in different places like the septum or the posterior wall. And then we can trace the whole ventricle and calculate the actual mass of the ventricle. So we'll look at wall thickness and LV mass. All 4 patients that have enough follow-up time at this point to be analyzed had improvements in the posterior wall thickness, ranging between 9 and 39%. And then 2 of the 4 have had decreases in the overall mass as well. In addition to these measurements, we are also seeing improvements in circulating biomarkers of heart stress, in particular troponin. Many of you have probably heard of troponin blood tests, but we are seeing consistent improvement in the troponin levels in those patients that have been followed the longest. So early days yet, but very promising initial signs. Certainly, again, a good reason to continue following the patients that have participated and enroll more patients next year. Next slide. So again, we're focused on adults, in this case with hypertrophic cardiomyopathy, clearly apparent on the pictures of the heart. Have to have an MYBPC3 mutation. You have to have symptoms, and again, low levels of neutralizing antibodies. And this study is based in the US, and we have a good geographic distribution of sites. Many patients do travel from different states to go to a study center. So if you're interested in learning more, please reach out to us at patient.advocacy@tenayathera.com, or you can visit our hcmstudies.com website. Okay, and the last slide, things to think about if you're considering participating in a gene therapy clinical trial. Next slide, please. Encourage everyone to learn as much as you can about a trial. It gets stuck on the last slide. Go figure. All right. But anyway, there can be frequent visits there, especially at the beginning of the trial. There's a 1-week hospitalization at the very beginning, just to be extra prudent about safety and monitoring. So clearly this needs to fit into the rest of your life. Think about what you expect in terms of the outcome of the trial. If you didn't have that outcome, how would you feel about it? It is a trial. We do not know whether these drugs will fully work or not. Do you understand the risks? Have you had all your questions answered? Have you weighed the potential benefits and the risks? How do you feel about the clinical team at the hospital center running the trial? And who is in your support network? So all important considerations. And now that the end here, the last slide is just our contact information. So thank you all for for joining this, for listening. I hope it was helpful, and I definitely look forward to answering your questions now.
Thank you, Whit. Greg Roof, Executive Director of the DCM Foundation, Genetic Cardiomyopathy Awareness Consortium. Again, I think we've got 15 questions so far, and they're coming in, so we're going to have a lot of questions. But I think overall, before we start that, again, want to thank you and Tenaya. We've known each other for a long time. Appreciate you guys being pioneers. I know patients with genetic cardiomyopathy appreciate it. Congratulations on the very positive news. And we'll get to the questions, but thank you very much. So let's start. I'm going to start with the first question and then work my way down. Zindi had first question is, I was told there is currently no plan to enroll patients without an ICD into the trial. I am on the cusp of receiving an ICD, but I'm in the UK and my EP is trying to hold out as long as possible. How likely is that I will be able to take part in the trial, or even if not, whether I could receive the therapy if it works? I also have children and would like very much to stop them experiencing what I do. So it seems like the real question there is sort of holding off on the ICD.
Yeah, well, thank you for your question. Thank you for considering the trial. We certainly, all patients need patients like you to consider participating in the trial so we can all learn more and advance therapy. I'm sorry to hear that your kids are carrying the gene mutation. We very much are trying to prevent disease for everyone, and you're not alone. Many of the patients participating in our trial are doing that because they know their kids are carrying the gene as well. Not your fault, nothing you could have done about that. Um, the PKP2 trial does require an ICD. That is going to be a part of the eligibility criteria for the foreseeable future for this trial for the next few patients, potentially up to the 15. But as we move through development and learn more about how TN-401 works, we anticipate treating earlier in the disease and treating people before they need an ICD, ideally to prevent the need for an ICD. The 201 trial is further along in that process. We started with that as a requirement to get the MYBPC3 gene therapy. That is not a requirement now in the trial. It turns out all the patients that are enrolled do have an ICD, but that's not a requirement now for 201 for the hypertrophic cardiomyopathy. But because of the more arrhythmic nature, the higher risk of events for ARVC, and because we use those ICD to measure and count events in order to get data to see how the drug is working, we will continue to require the ICD for now for that trial.
Perfect. Thank you very much. I'll grab this one, Whit, and if I'm wrong, please just stop me. So Andrew has a question. Any trials coming up for bag 3? I know of 3. I don't know in 2026. I don't know if they're formally approved or not, but I know a lot of them are applying for their IND to be able to start their clinical trials. So as I know, good news in the bag 3 world, we know of 3 potential clinical trials in 2026. From Michelle, for the cohort size, how is that determined? By regulating agency or by Tenaya, or by the number of patients who are eligible? Thank you.
Yeah, so it's by Tenaya. We make a proposal to the agencies, like the FDA or the MHRA in the UK. They can modify it as they see fit, but they accepted our proposals of 3 each for the initial cohorts, and we worked out with them that each of those 3 patients would be dosed 1 at a time and followed for safety for a short period before the next patient was dosed. That's how those initial cohorts worked. We're out of that phase now and into the expansion phases. So we monitor our progress, and we will continue dosing until we achieve the objectives of the trial, which are to understand the safety and tolerability, to be able to choose a dose for the next trial, and to look to see how the drugs are working, and where they're working most, and how we can best design the next trial. And for both of these gene therapies, it's possible that the next trial will be what we call the pivotal trial, the trial that finds the basis for approval so that the FDA can look at the data and approve the drug.
Great. Thank you. From Larry, he says, RE-ARVC, there are a number of clinical trials in progress with other companies. Ballpark on your part, how many years would you say till a remedy will be available? Also, are these remedies designed for everyone with ARVC or just those who are in severe progression?
Yeah, terrific. So it is great for patients and makes me happy that there are 3 different trials for PKP2 ARVC. The trials are going to be generally similar overall. but each uses a slightly different way to deliver the genes. They all use this vector called AAV, but they each use a different subtype of AAV. So we will learn in the near future here, in the next couple of years, potentially, which are the best, or do they all work? So far, the other two trials have released early data, and we're all seeing similar things in terms of early signs of improving electrical stability. Um, so, that'd be excellent if they all work. We will see. But a very exciting time for patients. Obviously, the more things that are studied, the more options that will make it through. In terms of the timing, we will partner with regulators on the design of those pivotal trials I mentioned before. That will really determine how long it will take to get the evidence that the regulators need for approval. So, it's not next year. It is gonna be measured, unfortunately, in years, but hopefully very few years.
Great. Thank you very much. And I'll try to take this, Whit, and again, correct me if I'm wrong. Any hope for upcoming research for DSP cardiomyopathy from an anonymous attendee? Again, what I know, there are about 35 different companies with genetic cardiomyopathy therapy pipelines. And I think about 81 is the last number I've heard as potential therapies. And a number of them are looking into different genetic cardiomyopathy genes. And so, I believe over time, most of all the genes will be covered. There is a lot of interest. And so I think for most of the genes, it's coming. Whether it's a particular gene therapy or group gene therapy, if the mechanism of disease is very similar. But I think for all types of cardiomyopathy genetic disease, I think that there is hope for the future. The timing on who comes out when is sort of up in the air. Whit, how much did I butcher that?
No, I share your enthusiasm, and we are really on the cusp of a variety of different genetic approaches. AAV-based gene replacement at the beginning, editing is a future thing. We have many different things coming into our armamentarium. DSP, definitely high unmet need. And we are working on that ourselves. I know other companies are. There are a couple of extra technical challenges with DSP that makes it take longer than PKP2. But we are trying.
Great. From Karen, could these gene therapies be used to treat lamin patients, effectively replacing the mutated gene with the normal lamin? So I guess we know there's a lamin clinical trial coming up in 2026. I don't know how public. I know it's public, but I won't name the company just yet. But so what do you think that the types of therapies you're doing could work on many of the genetic cardiomyopathy, in particular genes?
Yeah, each gene has its unique set of specific challenges that need to be solved. LMNA is another one that we are working on. Not ready for clinic, not ready for prime time yet. But again, what folks want is many companies working on it. We welcome progress coming from everybody. We all learn from each other. And I will say that while we compete, we know that success helps everyone, like including on the safety side. So we work together to maximize the safety, minimize the amount of immunosuppression required. We compare notes because the better we do for our patients, the better it is for everyone.
Great. Thank you. Another question about DSP. Could these treatments be used to address DSP as well? I think.
Yeah, just if DSP is longer, it's a longer gene spelled with more letters. It doesn't fit in a single AAV. And so that's the technical challenge. There's different approaches to solve that, and a lot of work being done. But that's why it's taken longer for DSP than PKP2.
Great. Thank you. Do patients in both trials require immunosuppression, and what type of immunosuppression is used and for how long?
Yeah, so AAV-based gene therapy generally requires immunosuppression for the IV delivered forms. And that's pretty much universal. So we use two drugs, prednisone and sirolimus. Some will use three drugs. They add, for example, rituximab onto that as well. It depends on the disease and the dose, how much you need. We some people will recognize those drugs that you have post-heart transplant, for example. The key difference is that this is temporary. So when the body has cleared the AAV proteins away, then there's nothing to stimulate the immune system anymore, and the immunosuppression isn't required. Um, as we've improved the immunosuppression, now we're at the point where the average person needs it for about 80 days. So it is immunosuppression. It's a short period, and it's an even shorter period with the higher doses. We taper it off during those 80 days.
Yeah. For everyone, heart transplant 4.5 years. I've been on immunosuppression for 4.5 years and will be for the rest of my life. Dealing with that and dealing with immunosuppression medications very successfully and from a long-term standpoint. Is the heart muscle thickening / inflexibility expected to reverse with TN201 for MYBPC3?
Yeah, so the most straightforward thing to do with a gene therapy is prevent the disease. The next is to stop the progression, and then the highest bar would be reversal. But in the animal studies, and we have animals that have, with the same gene mutation that have very similar increases in heart muscle hypertrophy, we do see reversal and improvement in heart function once the disease is established. That's what we're beginning to see, or we see early evidence of when we see the posterior wall thickness going down or coming back to a normal size wall and the mass index beginning to improve. So we are very much aiming to have reversal of disease once it's established.
Great. I'm going off script here. Question. And so it seems like in order for clinical trials in gene therapy, you're going to need people that are sick to see if there's improvement. If at some point the therapy works, it's approved the FDA, and then it can be then administered to patients, right? Many patients. At that point, would you say the key would be to try to give it to patients before the disease progresses too much, maybe the early signs of the disease? Or when do you see, and I know that's sort of up in the air and we're dreaming here, but when do you see, if and when a drug is approved for gene therapy for genetic cardiomyopathy, when will the most likely time patients, the optimal time for them to be dosed in their disease development?
Your vision is our vision, and many of the physicians that treat this disease, that's where they go right away to. This is really well suited to prevent disease. It's a one-time therapy. Once the gene is delivered to the heart cells, for a variety of reasons, it stays there. The key reason is heart cells actually don't divide. The cells are stable, so the gene is stable. So ultimately, getting this to patients before they even start having symptoms, that's a long-term vision. But the way development works and the nature of early trials, usually does involve the most severe patients that are not too late to respond at all. And those are the types of patients we're enrolling in our trials now. So think there hasn't been so much scarring of the ventricle that there aren't even enough myocytes left to respond. But as you've seen, the patients in the ARVC trial have gone through sometimes multiple ablations. In the MYBPC3 trial, have had myectomies and have continued to progress despite the myectomy. So, these are severely affected patients. That's why it's extra encouraging when we see signs of improvement or recovery. As we go forward and figure out how to maximize the safety and the efficacy of these drugs, we will move it earlier and earlier in the course of disease. Approval will be somewhere along that process. The first approval won't be for patients before symptoms, but that will be the long-term goal.
That's great. Thank you. And if it's okay, we might go 5 or 6 minutes over questions. I think we're halfway through 23 questions. So thank you for your questions.
That's terrific.
So Emily, hello, I'm a patient with ARVC. I'd be interested I would be interested in the trial. I know not everyone can be part of it. So, but after trial completion, what is the plan for getting this to patients? How much will it cost? And is this something insurance will cover? And thank you guys for doing this. So
Yeah, so one of our objectives for 2026 is to take this early data to the FDA and other health authorities in the world and map out exactly the answer to that question. I can't answer your question without aligning with the FDA and the health authorities. We also need to agree on what the plan is. But we're planning for the plan. We're meeting with them. We're taking, this data even from these first few patients who have chosen to participate and using that to think forward, and so we don't lose any time.
great, great. Thank you. From Kaylee, have you tried an intranasal delivery method instead of IV to bypass the liver?
Yeah, so no, we haven't tried that. We did try another way to bypass the liver is to administer it directly in the blood vessels that go to the heart rather than the peripheral intravenous. We actually did it into the veins that drain the heart, and we can make it go backwards up the veins and into the heart. We did that in animal studies. We have not done that in people. In our animal studies, it worked just as well, even with the liver in the system, worked just as well to give it IV, and that's certainly easier for patients. That said, there are some gene therapies that are delivered into the blood vessels of the heart, and it's working in those cases. So it just depends on the therapy that you're using.
Great.
It would be unlikely that if you inhaled it, it would get enough to the heart, just because getting through the nasal passages and across and into the blood vessels is not straightforward from the nose.
Perfect. Thank you. How do we know if we have a high percentage of AAV9 neutralizing antibodies?
Yeah, the only way to know is to directly measure it in a special blood test. Those aren't available, like you can't just go to your doctor and get that blood test. We built our own, and the different companies have sort of built their own tests, which we use. So it involves signing the informed consent form and getting screened for a trial. So in both our natural history studies and in the interventional trials, we do test for these so that patients can learn that way whether they have them. But there's not a convenient way.
Great. Here's a great question. Is there financial support for travel if one qualifies for enrollment?
Yes. Yeah. So
Great.
as I said, we've had patients, travel from different states to the study center, and the travel and lodging, it is covered.
Thank you. And very important. We appreciate that. Could you explain some of the risks that might be expected for both treatments? What are the potential risks? It seems like they were minimal, in your first cohorts, but what are some of the risks?
Yeah, so the key risks for our two gene therapies are just the reaction of the immune system to those viral proteins that we're using to deliver the gene. Those are there temporarily, but we do need the temporary immunosuppression. Without that, there would be a lot of liver damage. So it's essential, if you're getting a gene therapy, to be compliant with your immunosuppression regimen. So what we do is we start high, with a pretty high dose of prednisone. Well, not high compared to transplant necessarily, but a hefty dose, and then we gradually decrease that, a little bit every week to try to get it off. And again, we can do that in 80 days. But every week we measure the blood test for the liver to see if the liver is doing okay, and if there's a little bit of evidence of liver inflammation, we'll stop reducing the dose of prednisone. Sometimes we might go up a little bit. So that is happening in the trial. That's a kind of a frequent thing that we see a small bump in the blood test. And so we, keep at the current steroid dose or go up a little bit. But again, right now we are getting people off consistently in about 80 days time. Now, there are definitely theoretical risks, other types of immune reactions. that we haven't seen but have been seen in other gene therapy trials. So we monitor very closely. It could happen. So it is a theoretical risk for us, but certainly possible. And then, these are unknown therapies, so there could be a risk we haven't seen yet. So there's always the risk of the unknown in these. That said, AAV gene therapies have been used in hundreds of trials and treated thousands of patients. The type of AAV we use is called AAV9. That is the same one that's used in the huge success story called Zolgensma, which is a gene therapy for children with a disease called spinal muscular atrophy, or SMA. Thousands of children have been treated with AAV9 at this point. So we do have a extensive understanding of how the immune system reacts to AAV9 and how to best manage it.
Great. Thank you. Anything for the TPM mutations? Whit, I do not know of anything or anything I can publicly talk about. One of my 3 gene mutations is TPM1, but I do not know anything for the TPM mutations at this time. So,
Yeah, I don't have anything I can share either. Not one that we are working on today.
okay. TTM mutations are the most frequent genetic cause of DCM. Any pipeline studies related to this mutation? And then congratulations on the great success.
Yeah, TTN for Titan. Definitely a lot of work in the field, but I can't share specifics today, and not one that we are focused on in the near term here.
Yeah, I know there's I know there's potentially some things coming. I know there's people working on them, not that we can talk about, but I think that there is some hope there, and there is some hope in the future. I think and I think 2026 we can probably talk a bit more openly about that. So stay tuned. Join our webinar. For both programs, how much protein upregulation do you believe is required for meaningful improvements?
Yeah, so that will be determined by the studies. So we will start correlating the amount of protein that we're getting with the amount of clinical improvement. So in both cases, that we are seeing encouraging signs, improvements in the imaging, the heart, improvements of the electrical stability, improvements in New York Heart Association class. And we're doing that, with increases of 5 to 15% in protein. So that basically it's telling us that that could be enough to make a real difference. But we don't, there's not a good way to answer that question before doing this trial. So we are learning.
Get it. Thank you. Andrea, I know you had in one of your studies, you had 2 UK centers. Are you planning to recruit European centers in the near future? Thank you.
So yes, we're excited about the UK centers. We are very much looking forward to our first UK patient participating. For other countries, it's more likely that that will be in the next stage when we move on to the pivotal trials.
Great. There's a few more about, I think, DSP. Chris says, thanks, very exciting prospects. Will this be applicable to DSP? I think we've covered that. I think there are many more gene therapies coming for many types of genes.
I'll just say that, it's probably obvious from how we're seeing it, but each gene therapy is just for its gene.
Correct.
There are some examples where delivering one gene might help for people that have a loss of function in another gene. We're starting to see that, I think, in CPVT, for example, catecholaminergic polymorphic ventricular tachycardia. So there are ways that can work. You can cross genes, but most of these gene therapies are specific for people that have mutations in that gene.
Great. We've got time for one last question, then we've got to wrap up. Thanks, everyone. Might be a question or two, but one question for Bianca. Do patients continue to take their daily medication alongside the gene therapy during the trial?
Yes, that's the short answer. So in the case of ARVC, we expect patients to continue their antiarrhythmic medications, for example, certainly the beta blockers, if you're on flecainide. We ask that not be changed unless absolutely necessary during the trial. So if your dose changed, for example, that could change the PVC counts, and then we wouldn't be able to tell whether the gene therapy was working or not. So yes, continue standard of medications, your medications. Need to have them stable before you enter the trial and get dosed with the gene therapy. And then the hope is to maintain them stable unless there's absolutely needed to change it, so we can more accurately measure the effects of the gene therapy. The one exception here are the cardiac myosin inhibitors. So mavacamten is approved for obstructive hypertrophic cardiomyopathy. Only about 20 to 30% of MyBPC3 HCM patients have obstructive, but you can't be in our trial and on mavacamten.
Wonderful. Sorry, folks, we got a few more questions we couldn't get to. Whit, thank you so much. Dr. Tingley, I know you're battling a cold. You did wonderfully. Thanks so much for what you do and all the folks at Tenaya. Nikki, let's take us home.
Thank you so much, Greg. Thanks, Nikki.
Thank you. We'll have Justine bring our deck back up. So we want to certainly say a thank you to all of our sponsors whose logos you see on the screen. Their support allows us to present this important information. And thank you again to all the patient partner groups for their ongoing support. And next slide. So just a few final notes to wrap up. As stated earlier, you will receive an email with a recording of tonight's webinar. And I've jotted down a few notes to also include Tenaya's patient advocacy email and links to the clinical trial information in that follow-up email. Also, please register for our next webinar to be held January 21st, where we'll discuss understanding genetic testing results and the implications for you and your family. Registration is open and information can be found on the DCM Foundation and GCAC websites. And remember that even if you cannot attend live, just go ahead and register. And as a registrant, you'll receive a copy of the recording via email. Thank you, Justine, for your technical assistance behind the scenes. And thank you all for joining us. We're grateful you spent this time with us tonight and hope you leave feeling informed, supported, and empowered. Happy holidays, be well, and we'll see you soon.